All right, so we're going to talk about genes and heredity. It's going to be an introduction for you all. It is talking about some of the ideas about how genes work and how we are able to predict traits when plants or animals or whatever get it on, so to speak. How do we know what their offspring is going to look like? A lot of this work was figured out by Heredity Mendel. He was really the first to really systematically study heredity. That doesn't mean that obviously they had some idea, right? Because there's plant breeding going on and animal breeding going on long before Mendel. But Mendel was able to provide some insights, particularly by his systematically studying things, having an appreciation for really putting in a lot of math and working with the right organisms. The fact that he worked with pea plants and he can look at literally thousands of offspring provided insights that you can't easily do if you were to breed dogs, for instance. It'd be multiple generations. It'd take a long time. So pea plants was an excellent choice. And not only that, there was a lot of characteristics where he could find a trait that would breed true. And you can see a tall plant or a wrinkled seed or yellow seed and so forth. Those were some big advantages that he selected when working on pea plants, which he's famous for. He was a monk who studied science and mathematics and worked with garden peas in his monastery. So I guess one of the take-home messages is the foundations of science and biology can be studied in everything and anything. You know, a lot of times people have a bias, oh, he's just studying pea plants, or, you know, it's not human medicine or whatever. But realize understanding the foundations of genetics, we needed something like a pea plant and somebody passionate about studying it for us to appreciate how human genetics work. Not that humans should be the only reason for that. I mean, obviously, studying pea plants and being able to come up with food and different traits is important, right? Especially with modern agriculture. But anyway, Mendel was very critical for, or not critical, but essential for getting that off the ground. Now, the sad thing was that a lot of his work was ignored while he was alive. In fact, I think they really kind of rediscovered his work and found out how important it really was after his death. Again, the benefit of Mendel was that he chose garden peas. I don't know how, you know, a lot of it probably came down to just the fact that they're available and he had a garden, he could grow things in. But there was a lot of advantages for it. For instance, there's many distinctive traits, varieties that he can breed true. They're small and easy to grow, and they grow fast. So you can grow lots of different offspring really rapidly and look at the offspring afterwards, right? And look at their traits, what they look like. That's called the phenotype. He could actually control who was breeding, they could self-fertilize, but he could also take the anthers off, which would be the male reproductive parts, and fertilize another plant. So he could control who was breeding with who. So those were all big benefits because they have both male and female organs, and he could manipulate who was going to get the pollen, for instance, and then go down the carpel for the female part. So Mendel, for his research, selected 7 characteristics to study. And these all had distinct characteristics that were able to be contrast with one another. This is really important because these traits could then kind of be distinctive and breed true and not be necessarily connected or be independent of another trait. So in other words, you could have a tall plant with round seeds. Those would be independent traits from one another. That was a real benefit to the type of analysis he did. So it wasn't necessarily something that was connected together. Those were all very important things. So again, he was able to do a lot of self-fertilization. And so he could do many generations of a trait and then know that they bred true, that he could guarantee, in quotes, that the next generation would look just like the adult generation in height or round peas or color or so forth. Those were all big benefits, because there's a lot of traits. If we were to try to do these things with humans, like height, it's not just one gene, it's multiple genes, lots of different genes. So he was able to find distinct things in a pea plant, which was really good and helpful. That doesn't mean it's true for every, I mean, there are genes that in humans that would breed true and so forth. But again, this is something that was really beneficial in pea plants. So you'd have parental generations and parental generations that were pure bred lines. And then you could cross these pure bred lines of these different traits together. We call that cross-fertilization. And that would be the F1 generation, the first phyllo generation. Then you could cross that first generation with each other and have a second generation and see what might happen to those different traits. And you'll see that those helped him to start to understand that there's these different factors that were responsible for these different traits. Now, he didn't know about DNA and things like that. You know more about DNA than he does at that time. Chromosomes and things. That they weren't understood as being important, but he could at least extrapolate there was some type of factor. So here's kind of a typical Mendelian experiment, or Mendel's experiment. The cross-fertilizing two pure lines of plants, in this case, a purple line and a white flower line. They were self-fertilized for multiple generations and bred true. So these are the parental generations. Then he could swap the pollen from one flower to the next and have cross-fertilization take place. And then he noticed something kind of peculiar. The next generation, they were all purple. So he crossed white flowers with purple flowers, plants, and the next generation were all purple. That would be kind of like, that's strange. Does that mean the white flowers are all gone, that trait? I mean, how would he know? But it was peculiar, right? Did that just disappear? So what happened is in the next generation, the F2 generation, everything was self-fertilized, and lo and behold, the white trait was still there, the white flower trait and the three purple traits. Started realizing then that there was these traits never disappeared, and that some of these purple flowers are not pure lines of purple flowers. They must have an extra factor there. So these are the kind of things that he would start to think about and analyze in a very quantitative manner. So again, the first generation were pure lines, the parental generation, cross-fertilized those. He found them all to be purple, 100%. That's the phenotype, what they look like. The next generation, he bred them, and all of a sudden we had 3 purple types and a white type. He breeds this even a third time, and he sees, oh, wait a minute, the white trait is still there. And in fact, if you self-fertilize the white flowers, they stay white flowers. But that's not true with the purple flowers. There will be purple flowers that will still have the ability to pass on that white factor. Nowadays, we would call it genes, but you didn't know that at the time. So the second generation would have been a 3-to-1 ratio. So in other words, 25% of the flowers would have been white. And then later he sees a 1-to-1 ratio down here. So in other words, 2 of these flowers must have a white factor in there somewhere. You wouldn't necessarily know where though. And he did this over and over again for all sorts of different traits. You'd have a purple trait versus the white flower trait, yellow seeds versus green seeds, brown seeds, wrinkle seeds, green pods, yellow pods, so forth and so on. where he saw this ratio of 3:1. So the first generation, they would have, like the first generation, if you crossed yellow seeds with green seeds, they would all be yellow. And then the next generation, the F2 generation, we saw that 3:1 ratio again. So he made this statement that there was characters that had a dominant trait or dominant form and a recessive form. And again, that just means that it shows up more and it's expressed and shown in the next generation. It's not like dominant in the fight or something like that. People get kind of funny about these kind of terms once they throw a human perspective in it, if you ever noticed. But these are, like curly hair is dominant trait, actually. Does that mean that there's something special about it, or is it just a trait and how your hair gets kinkily, curly? The weird things, people think weird things sometimes when they put human perspective on it. It just means that this trait is what's being expressed and seen in the next generation. And so, but the trait never disappears. So in other words, the yellow seeds and the green seed plants are crossed. The next generation, they were all green, but the third generation, we start seeing green seeds again. Or sorry, when I crossed the yellow and the green, they were all yellow. And then the next generation, we see some green seeds again. So that's the whole point of that. But he doesn't know why, right? He just has a speculation. And so he calls these factors, and then he comes up with these ideas that they're independent factors, irrespective of, they're not destroying the other factor, all sorts of things kind of come up. And he puts together these ideas that still hold true today. So Mendel proposed a simple set of hypotheses. Remember, a hypothesis is a question that you can answer by measuring it, doing research, and having a result that you can either accept your hypothesis or not. And so he thought that parents do not transmit traits directly to their offspring, but via some type of factor. Now we would call these factors genes, but at the time he didn't know what those were, those factors. Each parent had 2 copies of these factors. He figured this out based on the ratios. Otherwise, if you only pass that single trait on, only purple, you'd only see purple from then on out. But the simple fact is that offspring must have had 2 factors, a purple and a white factor. And then when they self-fertilize, then we can see those traits again. So that's how he comes up with the idea that each parent has 2 copies of these factors. If they breed true, in other words, the white flower always breeds white flowers, then they have the same factor. And then we call that homozygous, same trait, essentially. Remember a zygote? We have a fertilized egg. If they have 2 factors that are different, like the purple flowers, they could then have offspring that are white or purple, we would call that heterozygous. And so the alternative forms of a factor lead to different traits. And so, again, we're talking genes again. If you have a purple gene and a white gene, they're the same gene, but they're different variety. We call that alleles. Does that make sense, everyone? So an allele is a different variety of the same gene. It can be found on the same part of the chromosome. Now, some other big terms that come up is the idea of phenotypes. What it looks like doesn't mean necessarily what factors it had or what genes it had, or in this case, alleles, or as we understand it. So a purple flower had both alleles. If it was heterozygous, it would have the purple and gene allele, purple allele, and the white allele. So the genotype is actually what is happening in the chromosomes and what's the actual genes. And sometimes you won't know that unless you do some type of molecular biology, or of course the way it was done back then and normally done, let's see what if they breed true or not. In the case of pea plants. The two alleles an individual has do not affect each other. So you either have the purple trait or, but you didn't necessarily have, so if you had a purple and a white trait, he speculated that you wouldn't necessarily have a light purple or something like that. Now we know it's more complicated, the phenotypes. But in his experiments, they always kind of bred true, so to speak. The presence of an allele does not ensure that that trait will be expressed in the individuals. In other words, again, you have the purple and the white alleles. The dominant trait was being expressed, but the white allele was not. Those were kind of things that he was putting together. Again, when we talk about alleles, we're talking about varieties of the same gene. So down here, we're looking at two different chromosomes. Well, actually, we're looking at the same chromosome in the sense, the fact that they're homologous chromosomes. But one came from the father and one came from the mother. And here is the gene locus. That is the location that's found on the chromosome. We can have around 1,000 genes per chromosome, by the way, or whatnot. And so this depends on the species. But here we have a gene for the purple trait. We give the big P to represent the dominant trait in the case of these flowers, which is the purple color. So we give it a big P. That's the dominant allele. And then you notice on this other trait that this plant hypothetically would have, it would be a heterozygous plant. You see a small p, which is the recessive trait. So it's found in the same location on two different chromosomes. And so they're independent of each other. Here's another chromosome in the case of another trait found on the same chromosome. Big Y, and in this case it's big Y and big Y, so there'd be a homozygous genotype. If it was a little y, it'd be a heterozygous genotype. So again, if these genes are found on different chromosomes, they're independent of each other. In this case, they're actually found on the same chromosome, so those traits aren't necessarily independent of each other because they're found on the same chromosome. But maybe I digress a little bit. The only thing we really care about at the moment was looking at these purple flowers and these white flowers and maybe figure out a way for us to figure out or predict what the next generation would be. And we do that through what we call a Punnett square. And that is by predicting what the gametes will look like. So remember, if you're a diploid organism, you have two chromosomes. And we've already talked about meiosis. What happens in meiosis? You have diploid cells, right, in your body, but your gametes are what? Haploid cells, right? So you have half the number of chromosomes. Your gametes are either sperm or egg, in the case of humans, right? Or other animals. The gametes are only going to have half the number of chromosomes. So what you do in a Punnett square essentially is you're saying what the gametes look like. So a Punnett square is a grid structure that enables us to predict, make a calculation of simple genetic crosses, not necessarily complex ones, but simple ones. And so here we have gametes coming from the purple flowers. In this case, they're all heterozygous. So we're talking about the F. In this case, we're talking about the plants that, after they had the parent generation that bred true, we had the offspring, they're all purple, they're all heterozygous. Then we're going to look at what's happening in the next generation. And so here we see the gametes. The adult plant would have been big P, little p, but they split off into making the pollen and so forth, the eggs or the ovum, and you have big P, little p. So in this case, let's say this is the father and this is the mother of this. In this case, the plant self-fertilizes and those gametes come back together and fuse from sexual reproduction and become fertilized. And now we have a plant with a seed with a big P, a seed with a big P, little p, and then another seed with a big P, little p, and then another seed with a little p. Then we just grow the plants and see what the traits look like. And what do you see here? That 3-to-1 ratio. 3 purple flowers and 1 white flower. If you look at the ratio, again, that's the phenotypes, you would have seen a 3-to-1 ratio. So what percentage of the next generation was white flowers? 1 and 1/4, right? Which represents 25%. So 25% were white flowers. 75% were purple flowers. So that gave us a prediction, a probability. And so here's again, how Mendel analyzed these flower colors. He's got, and you can do this for different traits. So here we have, the purebred white flowers, purebred purple flowers. We make a Punnett square based on the gametes that we predict. And then the next generation, they're all purple, 100%. Because we know that the big P is dominant. And when we have a recessive trait, we make it a small undercase letter. What if we breed a heterozygous, or we can find out it's heterozygous? Here we got a purple flower and another purple flower. We bring them together and we get that ratio again. And then we can breed these and see what happens in the next generation. So this can go on and on. So again, you can't know exactly what the genotype is by just looking at the phenotype. But if you breed the phenotypes, you can then, and you've done enough times, you can then know whether or not that trait was pure or not. You know what I'm saying? So if, in this example here, if we had a heterozygous purple flower, we know that, and then we breed it with a white flower, we see the next generation that half of them are white. We know that the parent had to be heterozygous genotype, even though the phenotype was purple. So that's our way. That's a way for us to figure out what the adult genotype would have been. But we wouldn't have known by just looking. Had all of them been purple, we would have known that this parent was homozygous purple by doing these breeding tests and experiments. Does that make sense? So you can use the Punnett square to reverse engineer and figure out what the parent type was if you got it from the wild or whatever. So Mendel's theory of heredity is one of the most important theories in the history of science. It's been well supported by experimental results that his major proposals are considered laws. Mendel's first law, or law of segregation, so this is the first law, and he didn't necessarily use it in the word of alleles and gene. This is the modern twist to it. 2 alleles of a gene separate when forming gametes, and gametes combine randomly, forming offspring. So 2 alleles of a gene, a single gene, so the 2 different traits of a gene separate when forming gametes, and then combine randomly in forming offspring. So that's why you have a probability. Mendel's second law, or law of independent assortment, alleles of a gene located on different chromosomes are inherited independently of one another. What that means is that if you go back to that picture, or this picture here, here is how, and I'll try to go back and explain that with the statements. The first thing is the formation. So here's the individuals we have up here at the top. We have a round yellow and a wrinkled green. They are homozygous in this particular example. So they're all round and all yellow. This is obviously the first generation, so they bred true. And then the wrinkled and green are the recessive traits. Then you put the gametes together, and here's what, in the case of these gametes, they're going to be all R, big Y, and all the gametes are going to be little r, little y, because that's the only possibility. We join them together, and all the individuals are all round and all yellow. So now we're seeing a heterozygous trait here. Does that make sense to everyone? So which ones are the homologous chromosomes? Well, these big chromosomes are the homologous chromosomes, right? These ones are homologous to each other here. Does that make sense? Homologous, the same. And then these big chromosomes are homologous to each other. And hopefully you remember meiosis, that ultimately this diploid trait becomes haploid. And so you have big R, big Y as a possible trait. Big R, little y is a possible trait. Little r, big Y is a possible trait, and little r, little y are possible traits. The point is these were all sorted independently of each other and all these different possibilities. In other words, this blue and this orange independently sorted and of course separated independently what was happening on these other chromosomes. Independent means they're not together. They're not influencing each other. So that's why you can have the big blue and the yellow together, or the big blue and the green together. They're independent of each other. Now, what happens if I had the yellow on this gene, or on this chromosome? Would it be independent at that point? No, right? Because they would be on the same chromosome. So while Mendel's laws of segregation and independent assortment are very important and really do happen, it gets a little more complex when you see the same alleles on the same chromosomes and a lot of other reasons. It doesn't mean that Mendelian genetics doesn't work, but it is a little more complicated. You didn't know about all the chromosomes and everything. But now, if you understand this rule, you can put it into a Punnett square. So here's a Punnett square of all the possible gametes from that last picture. This is just 2 genes. Could you imagine if you had to start looking at lots and lots of genes and try to get everything figured out? You can imagine this gets extremely complex, extremely fast, but it works well for simple traits. And then how persistent you are. But so what does this represent again? Big R, big Y represents a possible gamete. Because it was sorted, remember, a shuffling of cards. Here's another possibility from the F2 generation, because remember the F2 generation is this one. These are the possibilities. Because these gametes separated, and these genes separated through meiosis. Does that make sense, everybody? So what we're doing here is looking at all the possible gametes. And then we just lined them up, and then we see what the fertilized individual looks like, the diploid state. And then you're able to see what the possible genotypes are, and of course the phenotypes. And so if you look at what you're seeing here, the first row, they're all yellow. Why is that? They're all round. Why is that? Well, because remember, brown is the dominant trait, yellow is the dominant trait. So even when you see a little yellow, which would represent a green color, it's the recessive trait. It's only when you see a homozygous recessive trait that you actually see the color again. So again, these are the gametes. These are the sperm and the eggs. So I don't know, let's say I'm heterozygous. I don't know if it's this simple. Imagine I have curly hair. I've had some curly hair offspring. And I have a straight hair offspring. Of my 3 children, 1 was had straight hair, and 2 had curly hair. Does that mean that I don't know if it's this simple, maybe it's more than 1 gene, but that meant that I was probably heterozygous curly, right? Does that make sense? He can contest, I have a curly hair kid, right? Because my gametes put out curly sperm and straight hair sperm. Or pollen, let's keep it. I hope I'm trying to make, does that make sense a little bit? I had no idea that I had maybe a straight hair gene. Again, that's if it works exactly like that. So these are all the possibilities of seeing a trait. So maybe you have brown eyes. Brown eyes is a dominant trait, eye color trait. And then your kid has green eyes. That means you were probably heterozygous. Again, there may be more than a few different genes involved or alleles, but in general, there are some that's basically works that way. Everybody realizes that, right? So if you're pure brown eyes and you hook up the pure blue eyes, your offspring are probably going to be brown eyes. And then the following generation, you're going to have multiple different colored eyes, possibly, just like this peach plants. So your grandchildren will have different colored eyes, depending on who if they're heterozygous, they end up with a heterozygous brown eye. That makes sense to everybody? Works the same way, essentially. Granted, again, I'm oversimplifying the human traits, but this is how it works. This can work for different traits in animals too, this basic Punnett square. You just got to figure out all the different possible gametes. And of course, once you start talking 3 or 4 different traits at once, then you're talking about a really cumbersome Punnett square. I mean, imagine if you just threw another letter in for another trait on a different chromosome. Now we're talking several more possible gametes. Now, hopefully you got the gist about how genes work the other day in the previous lecture. This is just a quick recount of what I described the other day. That genes specify for amino acid sequences that make a protein. So genes are important for being the blueprints for a particular protein. Proteins determine in large measure what the body looks like and how it functions. And it's just a change in these mutations, a few changes that can make someone have curly hair versus straight hair. Might be just a little bit different. Make a hemoglobin a little different, and now you have sickle cell traits or sickle cell anemia versus normal red blood cells. It can be slight changes, few mutations. And now the protein's different. Because remember, genes are on DNA, on the chromosomes, and are the blueprints for making proteins. Evolution acts upon those phenotypes. And then you'll see those alleles. So again, here's our cell. Here's our nucleus. Here's the chromosomes in our nucleus. And then here is a chromosome. And then you'll see that a chromosome might contain 1,000 genes or more, or less, depending on, again, the species you're talking about. One gene, in this case, this gene they're talking about in this example, hemoglobin. Hemoglobin is the protein found in red blood cells, is found in the DNA. Again, it's going to be a gene on that chromosome. Then you remember how you make messenger RNA through transcription? If not, go back and check out that previous lecture where you unwind the DNA and you put the RNA in. That messenger RNA then hooks up with a ribosome, and we have what we call translation to make a chain of amino acids that ultimately make the protein. And then that protein, of course, is hemoglobin in this example, which hooks up in your red blood cells and carries the oxygen in your blood. A slight change in this gene, and now you have a hemoglobin that's slightly changed its shape, and now you have a red blood cell that's changed its cell shape, and now you have sickle cell. So again, that's what we're talking about. So when we're talking about these purple flowers and these white flowers, we're talking about proteins that were responsible for making these different traits in some manner, in some function, some way. Maybe the protein, was critical for making a pigment. Anyway, this is the first half of talking about Mendelian genetics. I will upload this, and then the second half, we're going to talk about some of the different things we see that aren't necessarily what we call Mendelian genetics. So that's what we'll talk about when we meet again at our next class. Anyway, thanks for listening, and please keep up with the homework and so forth, and have a good rest of the day. Take care.